Today's webinar will be given by Dr. Katie Parkins. Dr. Parkins is an application scientist here at Syntica Instrumentation, focusing a lot of her time on the optical imaging system that we offer from Vilber, and her time on the optical imaging system. She spent a lot of time working with the team at the Keenan Research Center as well. Katie holds a PhD from the University of Western Ontario in medical biophysics and completed a postdoctoral fellowship at Johns Hopkins University. Throughout her research training, Katie has focused on development and application of novel molecular imaging tools to monitor and treat disease, specifically focusing on cancer. Katie has used optical imaging extensively in her research and has been a tremendous resource to her lab mates and now our customers about how to truly get as much as you can out of optical imaging. Today, Katie will share with us much of that experience, trying to address many of the questions that she faced during her research, and which she still gets asked by many people looking to move into optical imaging or who are already using imaging in their work. She will then talk more about the Newton system, its features and capabilities, and finish up with a quick overview of a variety of applications in using the system. Should you think of any questions that you have during the webinar, please feel free to use the Q&A dialog box throughout the webinar, and I'll work through them with Katie at the end, filling the rest of the time that we have together. We do anticipate today's webinar to be around 40 to 45 minutes, leaving us plenty of time at the end to answer those questions. If we don't have time to answer all of the questions, Katie and I will work to pull together a transcript and get that out to all of the participants in the days and weeks ahead. Please also note that we are recording today's session, so if for some reason you lose connection or can't hear everything clearly, you will have access to the video file later for your review. Without further ado, I'm pleased to turn things over to Katie for today's webinar.
Great, thanks, Tanya, and welcome to everyone joining us for today's session. As Tanya mentioned, I'm going to start today, just in efforts of getting everyone on the same page with just a little review of preclinical optical imaging as a modality. We'll go through some of the common applications and the ways it's being used in research. And then I'm going to jump into the Newton 7 system. So we'll talk about some of the technical specifications, as well as some of the features that make the system unique. I do want to get into some of the ways or some of the considerations, I guess, that you would want to take into account if you wanted to add optical imaging to your lab. So I know many that are joining today do have some exposure already to optical imaging, but for those of you that don't, we do get quite a few questions in just how easy it is to add these capabilities to a non-imaging lab. So we'll cover some of those topics today. And then I'm going to finish just with a couple tips that I've learned from my research experience and how to get the most out of your optical imaging data. Okay, so to start off, for fluorescence imaging, following the administration of a fluorescent imaging agent, so either a fluorophore or fluorescent protein, an excitation light of appropriate wavelength is used to illuminate the subject, so either a mouse or a rat in this case. In the case of using a protein such as GFP, you would want to first engineer the cell line of interest to express GFP, or you could purchase a transgenic animal already expressing the reporter gene of interest, and then you would illuminate the subject. This then leads to excitation of the fluorophore or fluorescent protein and the subsequent emission of light. And so that light gets detected via a CCD camera above the subject, and then analyzed and converted into an image that will detail the location of the emitted photons or light from the subject. Now, similar to fluorescence imaging, BOI, or bioluminescence imaging, which we'll also talk about today, has been adopted by many researchers as a valuable preclinical imaging tool. Now, for BOI, it's quite similar. First, a small animal model needs to be developed that's capable of either expressing or secreting one of the luciferase enzymes. This could be a transgenic mouse or a mouse that's been injected with cells that can express or excrete luciferase. Second, an appropriate substrate needs to be administered to the animal. So this is the main difference between fluorescence and bioluminescence. This substrate will lead to the production of light from the enzymatic reaction of luciferase with the substrate. And then from here, it's very similar to fluorescence, where the light or the photons will get detected from the CCD camera above, and the image is produced. Now, keep in mind, unlike fluorescence imaging, bioluminescence doesn't need an external light source. So because of this, and because tissues don't exhibit endogenous bioluminescence, greater sensitivities and higher signal to background ratios can be achieved compared to fluorescence techniques. And we'll talk a bit more about this as we go on. Okay, so jumping into some of the common applications. Now for optical imaging, this list really does continue to grow. This is a modality that can be applied to almost any preclinical research lab. The most common, I would say, has been probably oncology. And this is just due to the fact that this modality is very easily applied for cell tracking purposes. So tracking where tumors go throughout the body, as well as monitoring tumor growth. It's also really great for groups that want to study whole body tumor burden, so metastasis studies, just because you can get that whole body image. Now, many don't know, but optical imaging can also be used to image the brain. So as a result, it's being used for lots of neurology studies. Biodistribution studies is another common one, again, because this is a whole body modality. So something like MR or CT, it might be quite expensive and time consuming to acquire images from the entire subject. Whereas in optical, quite simply, as you can see here on the right, we can get images of the entire subject. Treatment studies is another important one. Specifically, I want to mention bioluminescence imaging here, and that's because, as I mentioned, for bioluminescence, you need, for most of the reactions at least, depending on the luciferase you're using, you're going to need cofactors such as ATP or oxygen. And as a result, your signal is going to be representative of viable cells only. So that can be really important when you're looking at the effectiveness of a drug. Cell tracking, I mentioned earlier. And then ex vivo imaging, I would say this is becoming more popular. So groups that are looking to acquire images of maybe a hard to reach area, like a signal deep within a certain tissue, or maybe they're using a fluorophore, that has a hard time escaping the body. And in this case, ex vivo imaging can be used quite well to get signal. Okay, so jumping right into the Newton system. Now, the Newton system that I'm going to talk about today is manufactured by Vilber, which I'm sure many of you may not have heard of before. So Vilber is a leading life science company. Their headquarters and manufacturing plant are based in Paris, France, and they have regional offices in Germany and China. Now, they do have a presence in over 100 countries through a network of established distributors, and they've equipped more than 20,000 laboratories around the world. Now, I'm sure you're thinking, there's no way that there's 20,000 imaging systems worldwide, as many of you probably just recently became aware of the Newton system. And you're actually right. So Vilber was first created in the 1950s and started as a startup company manufacturing UV instruments. But by the 80s, they started imaging systems. So they released a gel documentation system for DNA, RNA, and protein gel imaging. And what actually ended up happening is they realized in about 2011, these publications started coming out where scientists were actually using these gel documentation systems to image animals due to their great sensitivity. And the images were actually quite high quality. And so from here, this inspired Vilber in 2016 to launch the Newton Mini. So this was the first animal imaging system. They basically maintained the high quality optics that were in their gel documentation systems, but just made some quick adjustments to allow it to be more feasible for animal work. So things like anesthesia and a heated stage. And then in 2019, Vilber formed a relationship with Cintiq as its North American distributor. And we now distribute the most recent model, which is the Newton 7, and that's what we'll be talking about today. And this system includes bioluminescence, fluorescence, as well as the most recent 3D optical tomography imaging. Now, in terms of the key technical features, There's a couple things that make the system unique. The first is that Vilber has developed this Dark 9 camera. So it's the latest generation of scientific CCD cameras, providing unrivaled level of sensitivity for bioluminescence and fluorescence applications. The camera is, as I said, a scientific 16-bit CCD camera. It generates up to 65,000 gray levels with a dynamic range of 4.8 orders of magnitude. So this just means that the very faint signals can be detected without saturating the strongest ones. Now, cameras generate quite a bit of heat, and heat will actually generate noise in the picture. And so the system does have camera cooling of about -90 degrees Celsius, and this is just to improve the signal to noise ratio. You'll find that this is something that's quite standard across the systems in the market today. I think one of the most important features of the Newton is the camera resolution. So it has camera resolution of about 4.6 megapixels. And I like to show these images here on the right because regardless of what the numbers say, you can see it just by looking at these two images. With the Newton image on the left, you can see it's very high definition, even just the bright field image. And the signal is quite localized. And then we have an image on the right that was acquired on a system that was 1 megapixel. So most of the competitor systems on the market today are close to about 1 megapixel. And you'll see the difference visually, it's quite pixelated. So this can make quite a big difference in terms of the quality of images that you're acquiring. Another unique feature is the lens aperture. So to our knowledge, the Newton system has the widest lens aperture on the market at about 0.7. And this is important because a wider lens aperture is going to allow us to collect more light, which improves the sensitivity. In terms of the darkroom itself, it's all motorized. So in a couple ways. So first, in the top video, you're seeing the stage itself move. So if you position the animal into the nose cone using anesthesia, you can actually pull, there's a little handle underneath those lights that you can pull the animal basically out, almost out of the entire system. So this can be really useful for groups that want to do things like IV injections so that you're not having to stick your head inside that dark box, trying to position yourself properly to get a good injection. You can actually just anesthetize the animal, have them on that heated stage, which is really going to help with tail vein injections, and then pull them out just for the injection. You'll also see in the bottom image that the LEDs move as well. And this is just so that we can scan the entire subject and keep the excitation consistent across the entire body. Now, I'm not showing it here, but you'll also see in the system, for those of you that are in Toronto, you'll see that the camera actually drops down vertically as well. And this just allows you to choose your field of view. So the closer the camera is to the subject, the sensitivity is increased. So depending on the tissue of interest that you're looking at or the size of field of view that you would like, you can adjust the camera vertically. The Newton has 8 excitation channels in order to cover the complete spectrum from blue illumination at 420 nanometers to 800 nanometers in the near spectrum. The powerful light generated by the spectral panels is categorized as laser class 2, so producing a monochromatic light, very narrow band illumination, and this will reduce the cross stimulation and increase the sensitivity of the images. It also has dual magnetron sputter-coated filter technology, and this is just going to ensure transmission above 90%, and very narrow band cutting for improved spectral separation and increased sensitivity again. Multi-spectral imaging is possible, so either using bioluminescence or fluorescence, and this would be used in order to detect more than one target at a time within the same animal. So for example, using more than one luciferase. So maybe you want to track a cancer cell line with firefly luciferase, and then therapeutic with Ranella luciferase. That can be done using different substrates. And then additionally, you could do fluorescence multispectral imaging, where you would look at using 2 of these dyes or fluorophores that I have here on the right to track different things. You just would want to be careful that you're using fluorophores that are far enough apart that there's not going to be any overlap. Okay, so I mentioned the 3D optical tomography earlier. So Vilber has developed a fairly accurate and inexpensive way of doing the optical tomography in this system. So there's these 3D near cameras on the side of the system that are actually going to acquire the topographic image of the animal to get the positioning. And then the main CCD camera above is going to acquire the multiple images of signal using emission filters from wavelengths in the spectral emission of whatever reporter gene is used. And then that signal will get reconstructed and positioned within the volume image of the animal. So this is what it ends up looking. So you're seeing a glimpse of the software here on the left. How this works is Vilber basically uses a 3D whole body mouse atlas. So this was generated using X-ray CT and cryosection data. So keep in mind, this is not a separate modality. So it's not optical X-ray or optical CT. It's not taking a new image of each subject. It's just going to pull in this atlas data and overlay your signal. But it's going to give you a fairly good indication of where that signal is within the mouse body. So you can see on the left, you can actually choose what you want to show. So you can choose to show the entire body of organs or bones, etc. Or you can just pull a couple things in at once just to get an idea of where that signal is coming within a certain volume. Now, Vilber is continuously updating the software tools for this feature, adding things in such as tumor segmentation. So here you can see you can actually segment out more than one tumor within the same animal. And then there's various measurement tools that you can try and get an estimation of both tumor volume as well as the distance between multiple signals within the animal. In terms of the software, again, this is continuously being updated, but for now it does, for those of you who have done imaging before, it's going to do, in my opinion, at least, it does everything that you have done in a previous optical imaging software. So for those of you that are familiar with the living image software, where you have the option of doing an automatic exposure, a manual exposure, or something called serial image or sequence image acquisition. So you could set up multiple images in a certain time frame. Maybe you want to take 10 images every minute with 30 seconds in between, and you can set that up so that you don't have to do each image individually. I think the most important thing to mention about the software is that it's non-proprietary. So this is really important, especially for those of you working in core imaging facilities, because this means that you can actually download the analysis software on your personal computer, and you don't have to actually be in the lab to analyze the images, which means that someone else can be imaging at the same time while you're on a different computer analyzing. Okay, so I just want to share a couple example images for those of you who are less familiar with the system. So on the left here, we have a luciferase expressing orthotopic lung met. And then on the right, we have an M-cherry expressing subcutaneous tumor. So both of these, at least the one on the left, because it's orthotopic, weren't visible to the naked eye. So it was deep within the lungs. And then the one on the right was just barely palpable. And then here we have a luciferase expressing subcutaneous hind limb tumor. Now, I mentioned ex vivo imaging earlier, and this is definitely an option. So for those of you who are interested, but are a little unsure in terms of the workflow or have questions about how that would work, I encourage you to reach out to me, and I'm happy to kind of walk you through. It is a little bit specific for each individual animal model. But just in general, I would say there's kind of two ways that you can do this. The first is that you would inject your substrate. In the case that you're doing bioluminescence, you would inject your substrate while the animal is still alive. You would allow a couple minutes for the substrate to circulate. And I even suggest taking an image in vivo first before you start the process, just to make sure that the substrate has got to the area that you're interested in imaging, and you can see a signal in vivo. If you can't see a signal in vivo, you likely will have a hard time seeing an image ex vivo. That being said, so if you see a signal in vivo, then you've given the substrate enough time to circulate. You can start to sacrifice the animal. So either isoflurane anesthesia probably works the best. And then I typically recommend doing the dissection in different levels. So start just by doing a vertical incision across the entire body and opening up just that initial layer of fur or skin. You'd be surprised just how much difference that initial layer can make. And then take images at each point. So slowly go a little bit further. So maybe you want to remove the intestines, etc., to get to different layers of tissues until you can see the signal that you're looking for. Now, some of you are going to have very difficult applications and that you won't see signal in vivo. And thus, when you start going through the initial top layers, you still will not see signal. And for those, I would recommend actually excising the organs like they've done here in this image. You can put them in a little petri dish, and you can actually just apply the substrate directly onto the tissue. And that's because the substrate itself should not give any background signal. So if you're just interested in knowing if there is signal at all, then that's definitely a good option. But keep in mind, it's going to be hard to control because the distribution is going to be kind of reliant on where you're delivering that substrate to. Okay, so now I'm going to jump into how to add optical imaging to your lab. So I commonly get this question. I think many people outside of the imaging world kind of believe that, you need to have a background in radiology or chemistry or tracer development to be able to, add imaging capabilities to your lab. And that's just not the case with optical imaging. It's very easy for really anyone to adapt this technology. And so what I want to start with is just the things that you would need if you were starting fresh and you knew really nothing about imaging. What would you need in terms of space and consumables, etc. So first is a lab bench allowing animal work. So here I'm showing the Newton system in the top right, just to give you an idea of the size of the system if you have not yet seen one. But keep in mind, this is pretty standard across the field in terms of size. And for the most part, the majority of the systems are benchtop systems that will sit on top of a bench, and they're fairly compact. So you don't need a lot of space. It's usually just the imaging box, which you're seeing on the left, and then a computer where you'll do your image acquisition and analysis. Other than that, you just need a substrate in the case of bioluminescence, and then cells expressing your relevant reporter or optical dyes. And so for those of you who aren't familiar with this, this is probably the point where you're getting uncomfortable, but I promise you I'm going to explain and how you can make this process simple. Okay, so I made this little chart. So for those that are new to optical, versus those that are more comfortable with optical. So I'd like to give everyone options. So first, if you're brand new to optical, I would suggest either starting with fluorescence or firefly luciferase BLI. The reason being, with fluorescence, you don't need a substrate. So that kind of takes away one, I guess, source of variability. Or firefly luciferase if you are set on doing bioluminescence imaging, just because it's probably the most common reporter gene used in the literature. So there's been lots of publications and kinetic studies, and we know a lot about this reaction. So it's a little bit easier to start out with. That being said, for those of you that are comfortable with optical, there's many different reporter genes to choose from, and I would encourage you to kind of do your research in terms of finding the best reporter gene for your application. For those that are new to optical, you can actually just buy substrate that's pre-made. So I would say that's probably what most groups do, just because it's not super expensive. And this is one way you can limit your variability. One supplier I'm familiar with is called ReadyJect. So for those of you that are interested, you can look into that, but there are many out there. And I know it gets a little bit confusing. For the most part, they should all be made the same if it's pre-made. And you can just buy it and store it in, typically you would store it in the freezer. And you can just, if it's the pre-made substrate, you can just thaw it 5 minutes before you're ready to image. And it's super easy. For those that are a little bit more comfortable with optical, there's many options to buy substrate in the powder form. So it may be a little bit cheaper. Again, the pre-made stuff is not crazy expensive, but it's a nice option if you're doing a lot of optical imaging to buy the powder form, and you can aliquot it yourself, especially if you're using a less common concentration. There's also many new substrates that are being made. As I said, the optical imaging field is constantly changing and improving. So lots of groups are making these new substrates and reporter genes for enhanced sensitivity. So if you're a little bit more comfortable with optical, I encourage you to look at those, especially for the more difficult applications. And similarly, you can order cell lines that are already stably expressing your reporter gene. So I mentioned in the last slide that you need either a cell line that's going to express your fluorophore or reporter gene, or you need an optical dye. So for those of you that don't have the infrastructure to make your own cell lines, don't worry, because you can actually buy these, and you can buy them a lot of places now. There's many options, especially for the groups doing cancer work. So I included this study just to give you guys some resources in terms of where you can go to look for these cell lines that are already engineered for you. So for those that are doing, I come from a background of cancer research, so I'm a little bit more familiar with these cell lines, but for those of you that are familiar with the ATCC, you can actually go to their website, and there is a long list of options. So for those of you that are doing cancer models, maybe the B16 or 4T1, these are common cell lines. They've already been engineered for you. And for the most part, they've already done the characterization work as well. So you don't even have to, worry that, are these really expressing what I think they're expressing? You can kind of skip all those steps. You can just purchase them, and then you can expand them out in your own lab for future use. So keep in mind this, if you're doing it properly, you can just get away with buying the cell line once and then expanding out for, years of studies. Okay, getting the most out of your imaging data. So this is kind of a summary of the most common questions that I get when people start diving into the imaging. Some of them are new, some of them have been imaging for a long time, and they're just a little confused. There's lots of groups out there that are doing things, their own way. They get comfortable with their workflow. So it's nice to kind of get everybody on the same page and understand what you should be doing with your data. So the first question I get a lot is, how long should I image for? So this one's really important. And I think the most important thing is to always run a pilot study if possible. I know it's not always possible, but try really hard to run a pilot study for your specific animal model to determine the kinetics. because it's going to look very different between disease models, the strain of mice you're using, time points for sure, and even subjects. So you may notice in your pilot study, you're imaging at day 0 and you have 5 animals, and those animals have what you think is the exact same disease state at the same time point and the same strain of mice. And their peak signal may look very, very different across the board. So it's really important to run a pilot study to try and predict what the time frame is going to be. I would say if you absolutely can't run a pilot, try to account for approximately ½ hour per mouse. This is assuming if you're using firefly luciferase or another luciferase. Fluorescence is obviously a little bit different. It may be, give or take, it may be a little bit more, it may be a little bit less, but if you're a user where you have to pre-book your time and pay ahead of time, I think it's always best to try and approximately ½ hour per mouse is probably going to get you in the ballpark of how much time you would need to accurately image. If it's possible, set up a sequence acquisition and then copy and paste your ROI or region of interest for each individual image. So that's because you want to make sure that you're catching the peak signal. So many people will actually just inject the substrate. They'll position the animal within the system. They'll get a nice image in the first 5 minutes and they'll say, okay, I have a great image for a paper. I'm going to move on to the next mouse. That is a little bit scary, especially considering if you're looking at, I mean, you don't want to ever do that, but it's especially scary if we think about, that data being used to study, the effectiveness of a drug, because you want to make sure you're using the same piece of data to compare over time. So for example, here we see if the substrate was injected IV, the peak signal is, about 5 minutes. Whereas if you're doing an IP injection, which most people probably are, if they're doing F-loop, the peak signal might not be till, just about 20 minutes. So you want to have a good idea of what this is. And initially, the best way to do this is copy and paste an ROI over the tumor or over the signal of interest, and then just use it for each additional image acquired and watch that signal go up over time. Image as frequently as needed until the peak signal is clearly passed. So I always suggest don't wait for, don't just stop imaging after the first decrease that you see after the peak signal, because sometimes the signal can look quite choppy like this. So if I had done that here, I probably would have stopped imaging after 13, 15 minutes, but you can see the real peak is actually here. So I think a good rule of thumb is wait to see that it's decreasing for about 2 to 3 images after the peak signal to make sure that it has clearly passed. Okay, how often to image? So again, pilot study is really important here. You need to consider how aggressive your disease model is. So for example, if you have a metastatic cancer cell line that's very, very aggressive, your timeline is going to be very different compared to a PDX model. So when I was doing my PhD, I was looking at a model that was so aggressive, the entire timeline from implantation to endpoint was 14 days. So because of that, and because of the data I wanted to get out of that, I would image every 2 to 3 days. Whereas another study I was on was a PDX model, and it took about 3 months for those tumors to even become palpable. So if I was imaging every 2 to 3 days for that model, it would have been so much imaging time and so much substrate wasted, to be honest, because the images are not going to look any different for a significant amount of that time. So keep in mind your disease model. And again, that's why the pilot studies are going to be important. Image as frequently as you can, but only if you're gaining new information. So obviously it's nice to have longitudinal data. So images from, any kind of biological changes, but if, for example, in the PDX model, like I said, if there's no change for months at a time, then it's really not going to benefit you or the study by imaging every couple days. So keep this in mind. If it's part of a treatment study, choose time points accordingly. So this is also important. You need to image directly before starting the drug to get the true pre-treatment measurement. So if you plan on administering the drug on day 15, you want to get a pre-treatment measurement, late day 14, if not the same day, would be ideal to make sure that you're getting an accurate representation of that pre-drug state. And then if you need to image more than once a week, which many of you will, I would always start with a pre-injection image. So this confuses people quite a bit because obviously there shouldn't be any circulating substrate. It's not like GAD, you shouldn't expect it to stick around. But keep in mind, it is possible, and it does actually happen. It's happened in my experience, and it will change depending on the health of your animal. So for example, at the beginning of a study, when your animal's quite healthy, you'll find maybe the substrate is clearing within, an hour or 2 hours. You go back to image and you can't see any signal because the substrate has cleared. Whereas at the end point of some disease states, the animal may not be healthy enough to clear it. And so you'll see a couple days later, I've seen it up to about 3 days later, some of the substrate was still there. So I would just be careful, make sure that if you are imaging that frequently, start with a pre-injection image just to be sure that the substrate from the previous session has been cleared and that's not contributing to your new signal. Okay, will animal fur interfere with the signal? The short answer is yes, but that's okay. There's lots of things you can do to try and limit this. So first off, always try and use nude mice when possible, just because then you don't have to worry about it. Obviously, that's not always the case because nude mice, of course, aren't going to have the full immune system intact. So many people will not want to use nude mice. They're also very expensive. But I would say if you have the option and it doesn't really make a difference to you, I would definitely suggest using nude mice so that you don't have to worry about the fur. If you cannot use nude mice, if it's not an option, white fur is a little bit better than black fur just because it will absorb and scatter less of the light. You can shave or nair the area of interest. So that's a pretty common technique you'll see here in the images I'm showing, that we shaved the area, and that really helps for any color of fur. But keep in mind, you need to try to avoid animal models that have any variability in skin color. So specifically those working with melanoma. So I had a collaboration before where we went to shave the animal or nair the animal, and it was the day of imaging, and underneath all that fur, even though the fur was the same color, the skin was actually almost like a calico, which really affected the signal in different places. So I think the most important thing across the board is to be consistent. So if you are going to shave or nair the area, just make sure that you're doing it for each imaging session, especially if you have a longer study. So if your study is, a month, you don't want to nair at the beginning of the study and then just let it grow back slowly for each time point. Make sure that it is it's done before each imaging session. Okay, if you're looking at detecting very few cells, or maybe have one of those more difficult applications, you're having a hard time finding any signal, there's a couple things that I like to suggest to try. This is not foolproof, so it's not going to work for everything, but there's a couple different things you can at least try to see if it will improve the sensitivity. The first is what I mentioned earlier, so the optimized reporters and substrates. So these are being developed all the time. You just have to look into the literature. And for those of you that have, a very specific application in mind that you think you may benefit from one of these, please reach out and I'm happy to suggest some that I know of. Injection site, so try and, again, this is very dependent on what you want to show, but if possible, inject as shallow as possible, because obviously the deeper that the signal is within the tissue, the harder it is going to be for that light to get out. Use nude mice if possible. Position the area of interest as close to the camera as possible. So for example, if the camera is over the animal and you're looking for lung signal, you're going to want to position the belly up, just like this. So here there's a mammary fat pad tumor. Similarly, it's very surface level. So I have positioned the animal facing up. If you were looking at image signal in the brain or maybe the spine, you would want that to be closest to the camera. So in that case, you would position the animal on its belly. And then similarly, the lungs, the abdomen, it really depends here, the specific organ you're looking at or the depth. But I would probably play around an image on in both positions to see what the signal is better at. You can increase the binning. So for those that don't know what binning is, it's basically the grouping of pixels in optical imaging. So if you're worried about sensitivity, you should set your binning to as large as possible. So typically this is 16 on most systems, and this will, it will lower your resolution, but it will increase or maximize your sensitivity. You can also increase your exposure time. So the longer your exposure time, the more photons that you can collect. So typically the max imaging time is about 5 minutes. I would suggest starting with a single source of signal to start off, just to simplify your results. So we'll get into this on the next slide, but you want to make sure that if you're looking to detect very few cells, you don't want to have a model like this where, you know, maybe these signals are limiting your ability to detect very few cells somewhere else. And then finally, you can adjust your field of view. So as I mentioned, the camera in the Newton system is going to be able to move vertically, and that's to be able to focus in on the area of interest. And the closer the camera is to the subject, that will improve your sensitivity. Okay, how to image multiple signals within the same animal or sample. So this is another important one that many people don't consider. If you have a strong signal, first let's start with just one signal. So here we have an animal with a mammary fat pad tumor, and it grows in a sort of logical fashion over time. It gets bigger as the tumor grows and the signal goes up. Now, this same mouse actually has signal in the lungs because this tumor has metastasized. We don't see any signal in the lungs here. And that's because this signal is so strong that it's masking the signal up here. So what you can do in this case is you can actually take either black construction paper or the Newton system comes with these separators that are made with a material specifically to block out the light. So you can actually use those to block out these stronger signals to see if there's weaker signals here that are being hidden. Okay, and then finally, I want to end off with help, my data looks weird, because this is probably the most common question I get. If it's even a question, it's more just, I guess, a scary thought when it comes to research. My first suggestion would be to take advantage of the whole body imaging modality. So there's so many imaging modalities out there where you have to choose this very small field of view to look for something very specific. Optical imaging is not like that. You can actually very easily get an image of the whole body. And so we should be taking advantage of that. And when I say that, I mean, there's lots of people out there that get concerned, and I was one of them for sure early on in my PhD. You would be looking for signal in the mammary fat pad, and all of a sudden you get signal in the brain, and you weren't looking for signal in the brain. So what do you do about it? And I think it's a really great way to kind of do the unexpected science where you follow the data rather than you're trying to answer the question that you set out to answer. And so I would encourage you to continue taking whole body images and just see if there's signal that you've been missing with other modalities, because that's one of the great benefits of optical. Understand what your signal means. So does your substrate require oxygen or ATP, etc.? So this is important too, because depending on the luciferase or the fluorophore that you're using, your signal could mean different things. So for example, in the case of a treatment study, if you have, maybe you have a tumor model that's going up over time, and then in the final week it goes down, and it's a control group, and it doesn't make any sense because this mouse didn't receive any treatment. And you need to consider, well, what does the signal really mean? And does it mean that there's less live cells? Because that may suggest that there's a treatment effect. Or maybe it means that there's necrosis. So if you get a tumor that's untreated that grows so big that you actually have like a necrotic core in the middle, that may cause your signal to decrease too. So try and understand what the signal means rather than just assuming that the signal is going to always go up with growth. Keep in mind molecular changes occur before anatomical changes. So that's related to what we just talked about. So the size of your tumor may not be representative of viable cells. And actually, in most cases, you'll see that it's not representative. So the size of the tumor will go up. If you're doing something like caliper measurements, it will continue to go up, but your BLI signal will often go up and peak and then go back down as you develop a necrotic core or scabbing or less viable cells over time. Try and always perform in vitro experiments to characterize each cell line that you're using. So if you are a group newer to imaging, as I mentioned, in some cases, depending on where you buy these cell lines, they will do the characterization for you. I do encourage you, especially if you're just getting the cell line, maybe from a collaborating lab or another scientific group, try and characterize on your own, even if it's just a simple experiment to look at cell number versus signal. And you should see a nice linear correlation between the two. Because if you don't want to go 4 weeks doing an in vivo study and not seeing anything, and then going backwards to try and characterize a cell line and realizing, maybe someone gave you a cell line that never even expressed luciferase, or the GFP cells weren't sorted, so there's only 10% GFP expression. So things like this, it's good to do it before you waste animals and before you waste lots of time imaging. And then as a last resort, if you're still getting some really wonky stuff and you've tried all of these various things and something is just not adding up, there are luminometer devices. So these are typically used for calibration when the system is installed. So they don't need to be used all the time because once they're calibrated once, they should be good to go. But this is always an option as a last resort to make sure that the system is reporting what you would expect it to. And with that, I'll pass it back to Tanya, and I'm happy to take any questions.
Great. Katie, thanks so much for the great discussion so far. For those of you listening in, if you have any questions, please, again, use the Q&A dialog box at the bottom of your screen, and I'll start working through those with Katie now. So Katie, you were talking about ex vivo imaging, and a really interesting question. Would using a fluorescence marker be more appropriate or easier than using luciferin, for example, so you didn't have to wait for the substrate to be delivered?
Yeah, that's a good question. Yes and no. So you are still going to find that using bioluminescence in general, you're going to get better sensitivity just because you're not going to get the background that you would get or the autofluorescence that you get in the case with fluorescence imaging. That being said, if you are set on using fluorescence models and you're not having any luck with in vivo imaging, ex vivo imaging is a great option because it will reduce, again, the layers, I guess, that the light has to go through to get to the CCD camera. So the images I showed earlier were actually ex vivo fluorescence images. So for whoever submitted that question, I'm happy to share that publication as well. And it will take you through kind of the workflow of how to go about doing that.
Perfect. You were also talking, obviously, throughout the presentation about different luciferases as well as different fluorophores. And I'm wondering if you, I guess, have your favorites and the ones that you've been most successful with.
So I would still say firefly luciferase, only because, as I said earlier, we know so much about it. There's very few surprises. There are better firefly luciferases than others. So for example, you will see variants that are available to purchase online that are more redshifted. And I have used those and they work really great. They still use the standard deluciferin substrate that any of the original firefly luciferases use. So they're a great option. Ranella luciferase, in my opinion, it's good. It's very sensitive. The problem is it requires an IV substrate injection. So it adds a little bit more variability because, at least for models where you don't know when you would expect signal, if you don't see signal, it gets tricky to know, was it the IV injection or is the tumor model not developed or the disease state not progressing yet? So I think that would be, my general opinion. There are some really great ones in terms of like secreted luciferase, which I don't know a lot about, but they're being more commonly used now for cancer studies because people can use them for blood reporters or urine reporters because they are secreted. So those are also good options, but I would say in general, I think I prefer to stick with firefly luciferase.
That makes sense. In the case where you were talking about imaging repeatedly and depending on the time frame between imaging sessions, if there was lingering signal or residual signal, is there any way to work around that? Can you subtract that signal out? How would you go about working with an animal in that situation?
So what I have done in the past, it really depends how flexible your timeline is. And this is why I always suggest doing a pilot study, because if you see it in a pilot study, you will almost definitely see it in the real study. But I would say if you are, doing the full study and you're surprised to see that there's lingering signal, I would encourage you to move your time point if at all possible, because your quantification becomes very messy if you need to subtract signal. That being said, you could potentially, depending on what the model is, like let's say you have multiple signals and maybe the signal that's lingering is not the area that you're interested in necessarily. You could cover it to see if that helps revive the new signal in the other area. But I would say rule of thumb, if at all possible, just move the time point because you don't want to image with remaining contrast agent.
Sure. Can you comment a little bit on the system's ability to do spectral unmixing to help with autofluorescence in the green channel, for example?
Yeah, so Vilber is working to further develop this, but there are a couple options already available in the software. So the first, I would say the easiest way that I like to use is the background subtraction. So this is kind of a less traditional way of spectral unmixing, but it really depends on your application. You can make an ROI for the background signal, and you can set this to wherever you wish, depending on where the background signal is coming from within your animal. And then you can draw your ROI around your desired signal, your region of interest, and do a simple subtraction. And that will take the background signal away from the other ROI. The other way is probably the more traditional way of spectral unmixing that people are more familiar with. And it will basically take a number of different images at various wavelengths to try and subtract out the background signal. So if you are looking at green, it will take, multiple images around that specific spectra to try and separate out the real signal or the true signal from any autofluorescence or background. And it will give you both the unmixed and the mixed images as an output.
Got it. Perfect. In your experience, and have you read any papers where you can use fluorescent bacteria and basically track or follow where they go in an infection model?
Yeah, absolutely. That's one of, I didn't mention it today actually, but that's one of the most common applications for optical imaging. So both with bioluminescence as well as fluorescence. And there's actually some bacterial luciferases that you can use. So for whoever has that question, again, reach out to me and I'm happy to share some of the resources I have on those specific applications, but definitely possible.
Perfect. Jumping back to the different fluorophores that you've had success with, can you comment on fluorescent fluorophores that you've used that you've had good success with, and perhaps comment on those that are less beneficial?
Yeah, for sure.
Thanks.
So anything further red is going to be the best option. No, I would say anything, over 600 is really ideal. M cherry is one that's more common. That's kind of in the middle. It's about 580, I think. But size 7, Alexa Fluor 750, DIR works really great. The ones that I would avoid, if at all possible, is GFP, TD Tomato, ZS Green. Those ones have a lot more autofluorescence, harder to see, especially at deeper tissues. So this isn't such an issue if you're looking at something very subcutaneous, not a huge concern. But as you start getting into deeper tissues, you definitely want to consider something as far red as possible, I would say.
Perfect. That makes sense. Jumping back to that lingering signal question, would it be possible to inject less substrate to have it be metabolized quicker and then be able to image more quickly? Is that something that would be advisable?
You could do that. That's an option. If you have an animal model where the animals are getting really sick, and you're seeing the lingering signal as a regular thing, you could potentially lower the dose that you're administering. What I would say is these are kind of precautions. It shouldn't be a common thing. So you shouldn't, every time you scan, there's no way you should be seeing lingering signal unless you have, one of these specific animal models that are very sick by the end. Right. So it's not something that everybody should, really be concerned about. And in that case, I would say try and stick with the concentration of substrate that's been used in the literature, just because it's being used for a reason, and that's for optimal sensitivity. And it should, in theory, be cleared within 24 hours. So I would stick with that unless, like I said, you're seeing lingering signal quite regularly. And then you could try changing things up and seeing if that helps.
Got it. Excellent. As we do kind of final remarks, if anyone has additional questions, please do still use the Q&A dialog box, because we will work through those questions and get answers off to you. But we have reached the 60-minute mark for today's session, so I do want to be respectful of everyone's time. So we will wrap things up. As we mentioned at the start of the webinar, we will be sure again to answer any of those questions over the next few days. I'd like to thank Katarina and Kelsey for their help in organizing today's webinar. And again, if you're in Toronto and you wish to speak with them about accessing the system at the Keenan Research Centre, please reach out to us and we'd be happy to connect you with them. Further, I would like to thank Katie for her presentation today, as well as for taking the time to address so many of your questions. And I trust that we have been able to provide you with some relevant information to your studies that you may be conducting back in your own labs. If in the days and weeks to come you have further questions, please feel free to reach out to Katie or myself, and we'd be happy to talk more about the Newton imaging system or any of the imaging systems that we have here at Cintiqa. We welcome the opportunity to discuss your research goals and if any of the equipment that we have could help move that forward. Thanks again for taking the time out of your day to attend, and we look forward to seeing you at a future Syntica instrumentation event.